Get ready to dive deep because today we're tackling the intricate world of eukaryotic gene regulation. We've got some seriously in-depth research here on how genes express themselves in these complex organisms, and I'm ready to really get down to the nuts and bolts.
You know, it's incredible how these processes actually govern everything. We're talking a cell's most basic functions, how it specializes, how it reacts to its environment.
So like the foundation of, well, everything, really.
Yeah,
Development, disease, the whole nine yards.
exactly. And we'll be covering a lot of ground.
Looks like we're diving into gene rearrangements, then chromosome assembly, then we'll get into the weeds with transcription factors.
Right. And then we'll wrap up with post-transcriptional regulation.
Seems like a packed agenda.
Definitely. We're going to start our deep dive at the very foundation, the genome level. We'll be looking at how changes within the DNA structure itself can have a fundamental impact on how genes are expressed.
Okay, let's start with gene rearrangements. How do those even work?
Well, the generation of antibody diversity is a perfect example. The ability of your immune system to recognize this incredible variety of antigens all comes down to a process called VDJ recombination.
VDJ recombination.
Right.
Okay, so how does that process actually create this huge diversity of antibodies?
So you have these specific enzymes, RG1 and RG2 recombinases. They cut and paste segments of immunoglobulin genes.
Okay, segments like what?
These segments are known as V, D, and J segments. And these enzymes target specific sequences flanking each segment. They're called recombination signal sequences.
So they're like little flags for these enzymes.
Exactly. These flags help facilitate the rearrangement process.
Okay, so what happens during this rearrangement?
Well, portions of the gene are deleted, which leads to unique variable regions being created in the antibody genes.
So each antibody gene ends up being slightly different.
Precisely. And that leads to a diverse repertoire of antibodies, each capable of binding to a specific antigen.
So it's like our bodies are shuffling a deck of genetic cards every time we encounter a new threat
Yeah.
to create like a custom-made defense.
Exactly. And this understanding of VDJ recombination has even paved the way for antibody-based therapies, where scientists can engineer antibodies to target specific diseases.
Wow. So we're seeing how something seemingly abstract at the DNA level has a direct impact on, well, real-world applications. Pretty incredible.
Absolutely. Now let's shift gears a little and talk about chromosome translocations. Have you ever heard of the Philadelphia chromosome?
Yeah, rings a bell. It's related to cancer, right?
Yes. The Philadelphia chromosome is a prime example of how gene rearrangement can lead to disease. It involves a piece of chromosome 9 breaking off. Chromosome 9 carries the ABL gene.
Okay. And where does it break off to?
This broken piece attaches to chromosome 22, right at the BCR gene.
So now you've got this fusion of two genes that are normally separate. What happens because of that?
Well, this fusion, we call it the BCR-ABL gene, produces a protein that's constantly signaling cells to divide. And this uncontrolled cell division leads to a type of cancer called chronic myelogenous leukemia, or CML.
So one misplaced gene segment can have some pretty devastating consequences.
Absolutely. And detecting the BCR-ABL fusion gene is now essential for diagnosing CML and figuring out the best treatment strategies.
Okay, so we've seen how gene rearrangements, whether they happen naturally or go wrong, can significantly impact our health. What's next on our journey through the genome?
Let's zoom out a bit and think about how DNA is actually organized inside the nucleus. DNA isn't just floating around in a jumbled mess.
Right, it's packaged in a very specific way.
Exactly. It starts with nucleosomes, where DNA wraps around these histone proteins.
Like thread on a spool.
Exactly. Then these nucleosomes form loops, and ultimately everything condenses into those iconic chromosomes we all know.
You know, I've always loved those chromosome pictures from, like, biology textbooks. They look so organized.
And that organization is crucial for gene regulation. The way DNA is packaged actually determines which genes are even accessible for transcription.
We call that differential gene accessibility, right?
That's right. So even though every cell in our body has the same DNA, the packaging can determine which genes are turned on or off in a specific cell type.
That's incredible. So like with red blood cells.
Exactly. Red blood cells need to make hemoglobin to carry oxygen. So the hemoglobin gene in these cells needs to be open and accessible.
Like an open book ready to be read.
Precisely. And the way that's achieved is by keeping that region of DNA in a relaxed, open chromatin state.
Allowing those transcription factors to come in and bind and initiate gene expression.
Exactly. And scientists have even developed techniques to study this accessibility, like the DNase 1 sensitivity assay.
Right. They use that enzyme, DNase 1, to identify regions of DNA that are actively being transcribed.
Precisely. Because those regions are more susceptible to being cut by DNase.
Okay, so the same genome can give rise to all these diverse cell functions simply by changing how DNA is packaged and accessed.
Exactly. It's remarkable.
It really is. All right, I think it's time to move up to the next level of gene regulation, transcription.
Transcriptional regulation is where things get really interesting. We're now focusing on the molecular machinery that controls that very first step in gene expression, transcribing DNA into RNA.
So we're moving from, like, the big picture of DNA organization to the nitty-gritty of how individual genes are switched on or off.
Precisely. And the first thing to understand is the sheer complexity of eukaryotic gene expression. Unlike prokaryotes, where you often have multiple genes transcribed together in a single RNA molecule.
Eukaryotic genes are mostly monocistronic.
Exactly, meaning each gene has its own individual on-off switch.
Okay, but it's not a simple on-off switch, is it?
Not at all.
Yeah.
Eukaryotic promoters, those regions of DNA where transcription begins, are incredibly complex. They're made up of multiple elements.
Like what kind of elements?
Well, first you have the core promoter. This region often includes something called the TATA box.
The TATA box? That's a familiar name.
Right. It acts as a binding site for RNA polymerase II, the enzyme that transcribes DNA into RNA, and also for general transcription factors, which help RNA polymerase bind to the promoter and initiate transcription.
So the core promoter is like the basic platform where all the transcription machinery comes together.
That's a good way to put it. But there's more. Upstream of the core promoter, you also have proximal control elements. These are sequences where regulatory transcription factors can bind.
And those transcription factors can either boost or suppress transcription, right?
Exactly. Think of them like dimmer switches that can fine-tune the level of gene expression.
Okay, that makes sense. And then we have the distal control elements, right?
Right. These elements can be located pretty far away from the gene they actually regulate.
Enhancers and silencers fall into that category, right?
Exactly.
So what makes these distal control elements so special?
Well, first of all, they're position and orientation independent.
Meaning it doesn't matter where they are or which way they're facing.
Precisely. They can even be located within introns.
Wow, so their location is very flexible.
Yes. And they can regulate multiple genes simultaneously. That's a key point.
Hold on, so these distant regions of DNA can control several genes at once. How is that even possible?
Through the wonder of DNA bending and looping, you see, certain proteins can bind to both the enhancer or silencer and to the promoter region.
Bringing those distant elements into close proximity, even if they're far apart on the linear DNA molecule.
Precisely. This facilitates the assembly of the transcription machinery and influences the rate at which the gene is transcribed.
So enhancers and silencers are like long-distance switches that can fine-tune gene expression from afar.
Exactly. And they play crucial roles in things like tissue-specific gene expression, how a cell responds to hormones, and even how a cell adapts to changes in its environment, like the heat shock response, for example.
It's like a complex communication network within the genome itself, with all these different elements talking to each other.
Absolutely. Now, let's delve into the world of transcription factors and how their activity is regulated.
Okay, I'm ready to unpack the world of transcription factors. Let's do it.
A key concept here is that it's not just about whether a transcription factor is present or absent. It's about how its activity is actually modulated.
So like a dimmer switch, it's not just on or off, but somewhere in between.
Exactly. And one way this is achieved is through specific synthesis. Different cells produce different sets of transcription factors.
So each cell type has its own unique toolbox of these regulatory proteins.
Exactly. And even within a cell, a transcription factor's activity can be further modulated through things like covalent modifications. Phosphorylation is a good example.
Right, adding a phosphate group to the protein.
Precisely. And that can either enhance or inhibit the transcription factor's ability to bind DNA or to interact with other proteins.
Okay, so it's like adding another layer of control.
Right. Then you have ligand binding. Some transcription factors need to bind to a specific molecule, like a hormone, to become active.
So hormones can directly influence gene expression by activating those specific transcription factors.
Absolutely. And then in eukaryotic cells, the nuclear membrane actually provides an additional layer of control.
How does that work?
Some transcription factors are kept out of the nucleus, sequestered in the cytoplasm.
So they can't even reach the DNA they're supposed to regulate.
Right, until a specific signal triggers their translocation into the nucleus. It's like a security checkpoint for the control center of gene expression.
So even the location of a transcription factor can dictate its activity. That's amazing.
And then there's dimerization. Many transcription factors actually function as dimers, meaning two protein subunits have to come together to bind DNA effectively.
It's like two halves of a key that combine to unlock a door, but in this case, it's gene activation.
Exactly. And all these regulatory mechanisms working together allow for incredible flexibility and precision in controlling gene expression.
It's mind-blowing how much is happening at the molecular level to keep our bodies running smoothly.
Now, let's delve into the mechanisms behind how transcription factors actually bind to DNA.
Yeah, we've been talking about these binding sites, but how does that interaction really work on a molecular level?
Well, DNA has that specific double helix structure. And running along that helix are grooves. Transcription factors have evolved these specialized structural motifs that fit snugly into these grooves.
It's like a lock and key.
Precisely. Each transcription factor's motif matches the shape of the DNA sequence it needs to bind to. And there are several common motifs: the helix-turn helix, the helix-loop helix, the leucine zipper, and the zinc finger.
That's a lot of different motifs. Are they all doing fundamentally different things?
While their structures can vary, the basic principle is the same. An alpha helix, which is a common structural element in proteins, fits into the major groove of the DNA double helix.
Enabling those specific interactions between the amino acids on the alpha helix and the DNA bases.
Exactly. That's how the transcription factor recognizes and binds to its target sequence.
So even though the motifs have different names, they all use an alpha helix to interact with the DNA.
Precisely. It's fascinating how evolution has come up with multiple solutions to the same problem.
Okay, that makes sense. Can we look at some specific examples of transcription factors to help solidify these concepts?
Absolutely. Let's talk about steroid hormone receptors. They're a great example of how ligand binding, subcellular localization, and gene regulation all come together.
Steroid hormones are involved in a ton of critical functions, right? I'm thinking glucocorticoids, estrogen, testosterone.
Exactly. They're involved in everything from development and metabolism to immune responses. And they exert their effects through these steroid hormone receptors.
So how do these receptors actually work?
Unlike many other transcription factors that reside in the nucleus, steroid hormone receptors start out in the cytoplasm.
So they're not even in the same compartment as the DNA they're going to regulate.
Right. Here's how it works. When a steroid hormone enters the cell, it binds to its receptor in the cytoplasm. And this binding causes a conformational change in the receptor, essentially activating it.
It's like a key that unlocks the receptor's potential.
The perfect analogy. And this activation allows the hormone receptor complex to move into the nucleus.
So the hormone is like a passport, letting the receptor through security and into the control center of the cell.
Exactly. And once inside the nucleus, this complex binds to specific DNA sequences.
We call those steroid hormone response elements.
Yeah, and they're located near the genes that the hormone is going to regulate.
So the hormone guides the receptor to the right spot on the DNA.
Precisely. And this binding event then kicks off the assembly of the transcription machinery, leading to either the activation or repression of those target genes.
It's like a chain reaction. The hormone sets everything in motion.
Exactly. It really demonstrates how interconnected these signaling pathways, cellular compartments, and gene regulation processes are.
Absolutely. Okay, we've covered a lot about transcription factors. Are there any other examples that might help us understand just how powerful and diverse these regulatory proteins are?
Let's talk about homeotic genes. These are master regulators that control the development of body segments and structures.
Homeotic genes. So we're talking body plans, right?
Yeah.
Like the basic layout of an organism.
Exactly. Think about the head, the thorax, the abdomen, the limbs. Homeotic genes provide the blueprint for all of that, determining which segments develop into what structures.
So they're like the architect of the body.
That's a great way to think about it. And when these genes are mutated, the results can be pretty dramatic. For example, in fruit flies, there are these famous mutations called the bithorax and antennapedia mutations.
Those mutations cause some pretty bizarre changes in the flies, don't they?
They do. In the bithorax mutation, flies actually develop an extra set of wings. So they end up with two thoraxes.
Two thoraxes. Wow.
And it happens because the bithorax gene, which normally represses wing development in a specific segment, is mutated. So wings can develop in that segment.
So one mutation can completely change the structure of the fly.
Exactly. And then there's the antennapedia mutation.
What happens in that one?
In that mutation, legs grow where the antenna should be. It's because the antennapedia gene, which is usually expressed in the thorax, is misexpressed in the head.
So instead of antenna, you get legs. That's wild.
It is. And what's even more fascinating is that these genes are evolutionarily conserved. Similar genes play crucial roles in human development, too.
So these tiny segments of DNA hold the instructions for incredibly complex processes.
Exactly. And with that, we've covered a significant chunk of transcriptional regulation. But our journey continues. There's a whole other layer of control that happens after transcription, post-transcriptional regulation.
Post-transcriptional. So what happens to that RNA after it's been transcribed? I kind of assumed it was a straight shot to becoming a protein.
It's not quite that simple. That RNA molecule undergoes a bunch of processing steps before it's actually ready to be translated into a protein.
Okay.
And each of these steps presents an opportunity for regulation, allowing the cell to fine-tune gene expression even further.
So it's like a series of checks and balances along the way
Yeah.
to make sure the right product is made at the right time.
Exactly. And one of the most fascinating aspects of post-transcriptional regulation is alternative splicing.
Alternative splicing, that's where different versions of a protein can be made from the same gene, right?
Precisely. So imagine you have a pre-mRNA molecule fresh off the transcription process. This molecule contains both exons and introns.
Exons are the coding regions,
Right, those are the parts that will actually get translated into protein.
and introns are the non-coding regions.
Exactly. And during splicing, those introns are removed and the exons are joined together to form the mature mRNA molecule.
Okay.
But here's the twist. The same pre-mRNA can actually be spliced in different ways.
Wait, so you can end up with multiple versions of an mRNA and therefore multiple versions of a protein, all coming from the same gene.
Precisely. And that's exactly what happens in alternative splicing. Different combinations of exons can be included or excluded, creating a whole bunch of different mRNA isoforms. And each isoform can then be translated into a slightly different version of the protein.
It's like having one set of ingredients, but being able to make different dishes with them depending on how you combine them.
A perfect analogy. This flexibility is what allows cells to generate a much wider range of proteins than the number of genes in their genome.
Wow. That's incredibly efficient. Can you give me an example of this in action?
Sure. Take the IgM antibody. This antibody is crucial for the immune response, and its function depends on whether it's membrane-bound or secreted.
Okay, that sounds like a pretty significant difference. How does alternative splicing control that?
The IgM gene has exons that code for a transmembrane domain. That's the part of the protein that anchors it to the cell membrane. So if those exons are included during splicing, you end up with a membrane-bound IgM antibody.
Makes sense. What if they're not included?
Well, if those exons are skipped during splicing, the resulting mRNA will produce a secreted IgM antibody, one that doesn't have that transmembrane domain.
Okay, so it's like choosing whether or not to include a specific attachment point.
Exactly. And that seemingly small change has a huge impact on the antibody's job.
How so?
Well, membrane-bound IgM acts as a receptor on B cells. It's what recognizes antigens and kicks off the immune response.
It's like a guard at the gate, sounding the alarm when it detects an intruder.
Perfect analogy. Meanwhile, secreted IgM circulates in the blood, binding to antigens and marking them for destruction by other parts of the immune system.
So it's like a mobile weapon, seeking out and neutralizing threats.
Exactly. And all this diversity of function comes from a single gene, thanks to alternative splicing.
Incredible. Okay, are there other post-transcriptional regulatory mechanisms we should explore?
Let's dive into the world of microRNAs, or mirror-airs. These are tiny regulators, non-coding RNA molecules.
Non-coding, so they don't get translated into proteins.
Right, but they can silence the expression of specific genes.
That's powerful. How do they do that?
It starts with transcription, just like any other RNA. An enzyme called RNA polymerase II transcribes a primary miRNA molecule, or pre-miRNA.
Good.
And this pre-miRNA has a specific stem loop structure. The mature miRNA sequence is embedded within that stem,
Right.
but the pre-miRNA needs some processing before it can go to work. An enzyme called drosha comes in and cleaves it, creating a precursor miRNA or pre-miRNA.
So like refining the raw material.
Exactly. Now, this pre-miRNA, still carrying that hairpin loop structure, gets exported out of the nucleus and into the cytoplasm.
Okay.
And in the cytoplasm, it encounters another enzyme called dicer.
What does Dicer do?
Dicer further processes the pre-miRNA, chopping it into a miRNA duplex. It's essentially creating a double-stranded RNA molecule.
Okay, so now we have two strands.
Right. But only one of those strands is going to become the active mirin. And that strand is loaded into a protein complex called RISC, the RNA-induced silencing complex.
RSC, got it.
And RISC uses that miRNA as a guide to find specific mRNAs that have a complementary sequence.
That's like a search and destroy mission.
Exactly. Once RISC finds a target mRNA, the miRNA can silence gene expression in a couple of ways.
Okay, how does that work?
If the miRNA matches perfectly with the target mRNA, RISC will just degrade the mRNA, destroy it completely.
So the mRNA is out of the picture.
Right. But if the match isn't perfect, RISC won't destroy the mRNA.
So what happens then?
It'll just block its translation. So the mRNA is still there, but it can't be used to make a protein.
So it's like silencing the message.
Precisely. And this level of control is critical for all sorts of cellular processes, from development to differentiation to responding to stress.
Okay, so miRNAs can fine-tune gene expression by either completely destroying an mRNA or just blocking its translation. It's like a dimmer switch for protein production.
Exactly, and they're involved in a wide range of processes.
It's amazing how much power these tiny RNA molecules have.
It is. And with that, we've covered a significant chunk of post-transcriptional regulation, but there's another critical aspect we need to discuss: protein degradation.
Protein degradation.
Yeah.
Okay, how does that tie into gene regulation?
Well, just like controlling the production of mRNA and protein is important, so is controlling their destruction.
Because a protein's lifespan can affect its overall activity.
Precisely. And one of the major pathways for protein degradation is the ubiquitin proteasome system.
Ubiquitin is a small protein, right?
You're right, and it acts as a signal for protein degradation.
Like a tag that says, destroy me.
Exactly. Ubiquitin gets attached to proteins through a series of steps involving these enzymes called E1, E2, and E3 ligases.
So they work together to label the protein for destruction.
Precisely. And often, multiple ubiquitin molecules are attached, forming a chain. This chain is the signal that tells the proteasome this protein needs to go.
And the proteasome is...
Think of it as a cellular garbage disposal. It recognizes that ubiquitin chain, unfolds the tagged protein, and then breaks it down into smaller peptides.
So it's like a recycling center for proteins.
In a way, yes. The cell can reuse those peptides to build new proteins.
It's incredible how much intricate machinery is involved in controlling the lifespan of a protein.
It really highlights how complex and elegant these cellular processes are. And with that, we've completed our deep dive into eukaryotic gene regulation.
Wow, that was a journey. We covered everything from rearranging DNA segments to chromosome packaging, and then we delved into the world of transcription factors and all those mechanisms of post-transcriptional control. It's amazing how much control is exerted at every single step to ensure that the right proteins are made at the right time.
Absolutely. And it's this incredibly intricate control that governs everything from a cell's basic functions to its specialized roles in a multicellular organism. It's the very foundation of development, of differentiation, and how a cell responds to its environment.
You know, we've explored how genes are turned on and off, but I'm curious, how do cells actually achieve that precise balance of proteins they need for specific functions?
That's a fantastic question, and it's one that scientists are still actively researching. It involves a complex interplay of multiple regulatory mechanisms, feedback loops, and signaling pathways, all working together to fine-tune gene expression.
Sounds like we have a lot more to explore.
We do! The world of gene regulation is vast and constantly evolving.
I'm looking forward to continuing our deep dive.
Let's continue our exploration in our next session. Picking up where we left off, I think it'd be interesting to look closer at how these regulatory mechanisms actually operate at the molecular level. You know, the interplay between these different layers of control is really fascinating.
I'm all for those aha moments, you
Yeah.
know, where seemingly separate ideas suddenly click together. So what kind of connections are we talking about here?
Well, we've talked about how chromatin structure, the packaging of DNA, can influence which genes are available for transcription. But a key part of that process is the actual covalent modifications of histone proteins. Those are the spools that DNA wraps around.
Right. We touched on how those histone modifications, things like acetylation and methylation, can actually change how accessible that DNA is to transcription factor.
Exactly. Specific enzymes can add or remove those chemical tags on the histones. It's like they're loosening or tightening their grip on the DNA.
So like HATs and HDACs.
Precisely. Histone acetyltransferases, or HATs, add acetyl groups to the histone tails. That neutralizes their positive charge and makes them less attracted to the negatively charged DNA.
So it relaxes the chromatin structure, making it easier for those transcription factors to get in there.
Exactly. And then you have histone deacetylases, HDACs.
Those remove the acetyl groups.
Right. And that restores that positive charge on the histone tails, making them bind more tightly to DNA. So the chromatin condenses and the DNA becomes less accessible.
So it's like a dynamic system of locks and keys, where those histone modifications control which parts of the DNA are open for business.
That's a great analogy. And what's even more fascinating is that these modifications can actually be influenced by a ton of different factors. Signaling pathways activated by hormones, environmental cues.
The cell is constantly getting input from its surroundings.
Exactly. It's like the cell is integrating information from multiple sources to decide how to adjust its gene expression.
Okay, I'm starting to see how histone modifications and chromatin structure are all intertwined. What other connections should we be making here?
Let's think back to our discussion of transcription factors.
Okay.
We talked about how they bind to those specific DNA sequences.
Right.
But their influence goes beyond just binding to DNA. Many transcription factors can also interact with other proteins.
They're forming complexes.
Exactly. And those complexes can either enhance or repress transcription. Some transcription factors can even recruit those enzymes that modify histones.
Creating a feedback loop.
Precisely. So for example, an activator protein might recruit a H8T to the promoter region, adding those acetyl groups and opening up the chromatin for transcription.
And a repressor could do the opposite, right?
Yeah.
Bring in an HDAC to silence things down.
Exactly. It's a beautifully orchestrated system.
Wow. It's like this intricate web of interactions with transcription factors acting as the masterminds, coordinating all these different events.
Exactly. And this complexity allows for an amazing level of fine-tuning in gene expression, making sure that genes are switched on or off at just the right time, in the right cells, at the right levels.
Okay. I'm starting to see how all these pieces fit together, but I have to admit it's a lot to keep track of. Any tips for how to make sense of this whole intricate regulatory landscape?
Focusing on specific examples can be really helpful. Let's consider a hypothetical scenario. A cell needs to respond to a stress signal, let's say heat shock.
Heat shock. Okay. That's when a cell gets exposed to high temperatures, right? And it triggers the production of those heat shock proteins to protect the cell.
Precisely. So when the cell senses that heat stress, it activates a signaling pathway, and that pathway eventually leads to the phosphorylation of a specific transcription factor.
Okay. And that transcription factor is?
It's called the heat shock factor, or HSF. And remember, phosphorylation is one of those covalent modifications that can change a protein's activity.
Right. It can act like a switch, turning a protein on or off.
Exactly. In this case, phosphorylation activates HSF. It allows it to bind to its target DNA sequences,
Which are located near the genes for those heat shock proteins.
Precisely. So the activated HSF binds to the DNA and triggers the transcription of those heat shock protein genes.
So HSF is like the emergency response team, getting those protective proteins made quickly.
Exactly. But there's another layer to this. HSF can also interact with those enzymes that acetylate histones, particularly in the regions around those heat shock protein genes.
So it's making the DNA even more accessible for transcription.
Precisely. So HSF isn't just directly activating transcription by binding to DNA, it's also indirectly boosting transcription by promoting that histone acetylation. It's like hitting the gas pedal twice.
Wow. That's a great example of how those different regulatory mechanisms can work together to create a very specific cellular response. It's like seeing how all the different parts of an orchestra work together to create a symphony.
It truly is. And with that, we've gained a deeper appreciation for how those regulatory mechanisms control eukaryotic gene expression. But our journey isn't over. We've mostly focused on how genes are activated, but it's just as important to understand how they're turned off.
Gene silencing. It sounds just as important as activation. I mean, why is turning genes off so crucial?
Well, imagine a cell constantly churning out proteins it doesn't need. That would be incredibly wasteful, and it could even lead to harmful imbalances. So gene silencing mechanisms give cells the ability to fine-tune gene expression, making sure that proteins are only made when and where they're actually needed.
It's like having a brake pedal to counter that gas pedal of gene activation.
Exactly. And just like with activation, there are multiple layers of control involved in silencing genes.
Okay, let's dive into those silencing mechanisms. Where do we start?
Let's go back to chromatin structure. Remember how we talked about open chromatin being permissive for gene expression?
Right.
Well, now let's think about the opposite scenario. Tightly packed chromatin, what we call heterochromatin.
Heterochromatin, that's like the silent, mysterious part of the genome, right?
You got it. Heterochromatin is densely packed DNA, and it's mostly inaccessible to the transcription machinery.
So genes located in heterochromatin are essentially silenced.
Precisely. And this type of silencing is often stable and heritable, meaning it can be passed on to daughter cells when the cell divides.
So it's like a long-term strategy for gene silencing, making sure that certain genes stay off for generations of cells.
Exactly. And this is especially important for things like maintaining a cell's identity and preventing the expression of genes that could interfere with its specialized function.
It would be pretty disastrous if a heart cell suddenly started expressing genes for digestive enzymes.
Precisely. Now, heterochromatin formation is a powerful silencing mechanism, but it's not the only one at play. Remember those tiny regulators we talked about earlier, the microRNAs?
Those are the ones that can target mRNAs and silence their expression, right?
Exactly. We talked about how miRNAs can either degrade those mRNAs or just block their translation. And that silencing effect is incredibly specific. It only targets mRNAs with a complementary sequence.
So it's like a targeted strike, silencing the expression of a particular gene.
Precisely. And that precision makes miRNAs incredibly versatile tools for fine-tuning gene expression across a whole bunch of different cellular processes.
Okay, so we've got heterochromatin formation and miRNA-mediated silencing. What other silencing mechanisms should we be aware of?
Well, let's not forget about DNA methylation.
Right, that's when a methyl group is added to a cytosine base in the DNA.
You got it. And that seemingly small chemical change can have a big impact on gene expression. Well, generally speaking, DNA methylation is associated with gene silencing. If a gene's promoter region is heavily methylated, it becomes difficult for transcription factors to bind.
So it's like putting up a do not disturb sign on that gene's control panel.
A great analogy. And what's even more interesting is that environmental factors, things like diet or stress, can actually influence those DNA methylation patterns. It's another example of how our environment and our genes are constantly interacting.
It's pretty amazing to think that our experiences can actually change how our DNA is modified, and that can potentially affect gene expression.
It is. It really highlights the dynamic interplay between nature and nurture.
Okay, we've explored a variety of silencing mechanisms. The long-term stability of heterochromatin, the targeted precision of miRNAs, and the sensitivity of DNA methylation to the environment. It's incredible how much control is exerted to ensure that genes are turned off at the right times and in the right places.
It is. This intricate control is crucial for maintaining what we call cellular homeostasis. It makes sure that cells function properly and can respond appropriately to their ever-changing environment.
I'm starting to understand that gene regulation isn't just a simple on-off switch. It's more like a balancing act.
Yeah.
This constant tug of war between activation and silencing mechanisms, where the outcome determines the ultimate level of gene expression.
That's a perfect analogy, and that dynamic interplay is what allows cells to be so adaptable, to maintain their identity, and to carry out their specialized functions.
All right. We've delved into gene rearrangements and chromosome assembly, explored the complexity of transcription factors, and uncovered all those different mechanisms of post-transcriptional regulation, including the importance of gene silencing. But I have a feeling there's still more to our deep dive.
You're absolutely right. We've mainly focused on the molecular mechanisms of gene regulation. But there's another fascinating aspect to consider: the spatial organization of the genome within the nucleus.
Spatial organization. So we're talking about how DNA is arranged in three-dimensional space. I thought we already covered that when we talked about chromatin structure.
Well, chromatin structure gives us a framework for understanding DNA packaging, but it's only part of the story. Newer research is showing that the genome isn't just randomly distributed within the nucleus.
There's a higher level of organization beyond just how DNA is packaged locally.
Exactly. It's organized in a very specific and dynamic way. Certain regions of the genome preferentially interact with each other and with certain nuclear structures.
Okay, so like different neighborhoods within the nucleus, different areas specializing in different functions. How does that impact gene expression?
A good example is the nucleolus. That's the prominent structure within the nucleus where ribosomes are assembled.
Right. The ribosomes are the protein synthesis machines.
Precisely. And certain regions of the genome, particularly those involved in making ribosomes, are actually located near the nucleolus.
So it's like having the protein production factories located near the raw materials.
Exactly. And this close proximity probably makes ribosome assembly much more efficient. It also ensures those genes are readily available when needed.
Makes sense. Are there any other examples of these specialized neighborhoods within the nucleus?
Another interesting idea is the formation of what are called transcription factories.
Transcription factories, what are those?
These are specialized areas within the nucleus where multiple genes are brought together for coordinated transcription, and these genes are often involved in similar functions.
So it's like an assembly line for gene expression.
Exactly. And what's really amazing is that the makeup of these factories can change dynamically depending on what the cell needs.
It's a flexible system.
Precisely. And that flexibility allows the cell to fine-tune its transcriptional output. So, for example, if the cell needs to respond to a specific signal, it can rearrange those transcription factories to prioritize the expression of certain genes.
That's fascinating. It's like the cell has this incredible ability to reorganize its workspace to optimize efficiency.
Exactly. And this spatial organization adds yet another layer of complexity and control to gene expression. It's like a city planner who designs a city to maximize its functionality.
I'm starting to appreciate how this spatial organization of the genome fits into the bigger picture of gene regulation.
It's all about creating a system that's both robust and adaptable.
We've explored a huge range of regulatory mechanisms, from the intricacies of transcription factors and all those post-transcriptional modifications to the spatial organization of the genome within the nucleus. It's been a fascinating journey, but I have a feeling there's still more to uncover.
You're absolutely right. We focus on the mechanisms that control individual genes, but there's a whole other level of regulation that controls the expression of entire chromosomes.
Entire chromosomes, wow. What kind of mechanisms are we talking about here?
One really fascinating example is X chromosome inactivation in female mammals.
X chromosome inactivation, that has something to do with dosage compensation, right?
Exactly. Female mammals have two X chromosomes, while males only have one. So to balance out that difference in gene dosage, one of the two X chromosomes in females is randomly inactivated early in development.
It's like silencing an entire library to make sure that only one copy of the books is being read.
A great analogy. And this silencing is incredibly stable. It lasts throughout the organism's entire lifetime.
Wow. So how does this inactivation actually happen?
It's a multi-step process, but a key player is a long non-coding RNA called ZIST. ZIST is produced from the X chromosome that's going to be inactivated, and it essentially coats that chromosome, attracting proteins that modify chromatin structure and silence gene expression.
So it's like draping a silencing blanket over the entire chromosome.
Precisely. And this is a great example of how entire chromosomes can be regulated as units, making sure that gene expression is balanced and coordinated across the entire genome.
It's really a testament to the power and precision of these regulatory mechanisms.
Absolutely. It really highlights how gene regulation is a multifaceted process operating at many different levels.
We've explored gene regulation from the molecular level to the chromosomal level, uncovering a whole array of mechanisms that control when, where, and how genes are expressed. But I feel like there's one more layer we need to peel back: the impact of the environment on gene expression.
You're absolutely right. We've touched on the concept of environmental influences throughout our discussion, but it's worth emphasizing that genes don't function in a vacuum. They're constantly interacting with their surroundings, and that interplay can have a profound impact on how those genes are expressed.
So our experiences can actually change how our genes are expressed. That's a pretty powerful concept.
It is. Think about identical twins. They share the same DNA, but their life experiences can shape their health and development in very different ways. And this is partly due to what we call epigenetic modifications. Those are changes that can alter gene expression without actually changing the underlying DNA sequence.
Epigenetics. That's like adding annotations to the genetic code without changing the code itself.
A perfect analogy. And one of the key mechanisms involved in epigenetics is DNA methylation, which we talked about earlier in the context of gene silencing.
Right. Adding those methyl groups to the DNA can effectively switch genes off.
Exactly. And what's fascinating is that environmental factors, things like diet, stress, exposure to toxins, can actually influence those DNA methylation patterns.
So the choices we make, the experiences we have, those can actually change how our genes are expressed.
Absolutely. For example, there are studies that have shown that a mother's diet during pregnancy can actually affect the DNA methylation patterns in her offspring. And that can potentially have long-term consequences for their health.
Wow. So our experiences can leave a lasting mark on our gene expression, even across generations.
Exactly. It really underscores how intertwined our genes and our environment truly are.
We've journeyed from the microscopic world of DNA and transcription factors to the grand scale of chromosomal regulation and the profound influence of the environment. It's been an amazing exploration, but I have a feeling our deep dive isn't quite over yet.
You're right. We focus primarily on the nucleus, the control center of gene expression. But gene regulation doesn't stop with transcription. There's a whole other world of regulatory mechanisms that operate at the level of RNA. These mechanisms influence how, when, and where proteins are actually synthesized.
RNA regulation. Okay, what kind of mechanisms are we talking about here?
Let's go back to alternative splicing for a moment. Remember how we discussed that process? How it allows cells to create multiple protein isoforms from a single gene by selectively including or excluding those exons during RNA processing.
Right, like creating different outfits from the same set of clothes by mixing and matching different pieces.
That's a great analogy. And what's fascinating is that alternative splicing itself is regulated by a variety of factors, including signaling pathways, developmental cues, and even environmental stresses.
So it's not just a random process. The cell can actually control which splice variants are produced.
Absolutely. For example, certain hormones can activate signaling pathways that change the activity of splicing factors. Those are the proteins that orchestrate the splicing process. And that can change the relative abundance of different splice isoforms, fine-tuning the cell's proteome in response to specific signals.
Okay, so the cell can adjust its protein repertoire to suit the situation.
Precisely. And this flexibility is crucial for an incredible range of cellular processes, from development and differentiation to responding to environmental challenges.
Okay, so alternative splicing gives us this way to regulate gene expression at the RNA level. Are there other RNA-based regulatory mechanisms we should be aware of?
Absolutely. Let's talk about RNA editing.
RNA editing. That sounds intriguing. What exactly is it?
It's a process that actually changes the nucleotide sequence of an RNA molecule after it's been transcribed from DNA.
Wait, so the RNA sequence can be changed after transcription? Doesn't that kind of go against the central dogma of molecular biology?
It might seem counterintuitive, but it's a real phenomenon. And while it's not as widespread as alternative splicing, RNA editing can significantly impact gene expression.
How so?
Well, by changing the sequence of an RNA molecule, you can actually change the amino acid sequence of the protein that it encodes. So you're essentially creating a new version of the protein, one that could have different functions.
So it's like making a last-minute edit to the protein's blueprint.
Exactly. And this editing process is incredibly precise, targeting very specific nucleotides within that RNA molecule. There are two main types of RNA editing that we see.
Okay, what are they?
The most common one in mammals is adenosine to inosine editing, or A to I editing.
A to I editing.
Right. And then there's cytidine to uridine editing, or CDU editing. That one's less common, but it's still found in a variety of organisms.
Okay. Do you have any examples of how this RNA editing can affect gene expression?
Let's look at the apolipoprotein B gene. This gene plays a crucial role in lipid transport.
Apolipoprotein B, that's involved in cholesterol metabolism, right?
Exactly. And in humans, this gene actually produces two different protein isoforms, ApoB100 and ApoB48.
Two isoforms from the same gene, so alternative splicing again.
Actually, in this case, it's RNA editing that creates that shorter isoform, ApoB48.
Okay, so how does that happen?
A specific enzyme called ApoBec1 comes into play.
ApoBec1.
And this enzyme deaminates a specific cytosine base in the apolipoprotein B mRNA.
Deamination. That's removing an amino group from the molecule.
You got it. And this seemingly small change from cytosine to uracil has a major impact on translation.
How so?
Well, that edited codon, which originally coded for the amino acid glutamine, now becomes a stop codon.
A stop codon. So that tells the ribosome to stop translation.
Exactly. So you end up with a shorter protein, that ApoB48 isoform.
So RNA editing is like creating a shortcut in the protein's blueprint, resulting in a truncated version.
Precisely. And this is crucial for lipid metabolism. ApoB100, the full-length isoform, is what transports LDL cholesterol. That's the bad cholesterol.
Right. High LDL levels can lead to heart disease.
Exactly. But ApoB48, that shorter isoform, is involved in absorbing dietary fats and cholesterol. So, thanks to RNA editing, you have these two specialized versions of the protein, each playing a distinct role in lipid metabolism.
That's amazing. It really highlights how RNA editing can create protein diversity and fine-tune gene expression in ways that just wouldn't be possible with alternative splicing alone.
Exactly. It adds another layer of complexity to the process.
I'm really starting to appreciate the flexibility and power of these RNA-based regulatory mechanisms.
They're essential for making sure the right proteins are made in the right amounts at the right time in the right place.
Okay, we've explored gene regulation from the molecular level to the chromosomal level, uncovered those mechanisms that control when, where, and how genes are expressed. We've even delved into this world of RNA-based regulation. But I have a hunch there's still more to our deep dive.
You're right. We focus primarily on regulating those protein-coding genes, but a significant portion of the genome doesn't actually code for proteins at all.
That's what used to be called junk DNA, right?
hmm
But I know scientists are learning that it's not junk at all.
Precisely. And one of the most exciting recent discoveries has been the identification of a whole host of non-coding RNAs, or ncRNAs. These ncRNAs are playing critical roles in regulating gene expression.
Non-coding RNAs, so they don't get translated into proteins.
That's right. But even though they don't code for proteins themselves, they can have a significant impact on the expression of protein-coding genes.
Okay, that's interesting. But how do they exert their influence if they're not becoming proteins?
Well, ncRNAs come in a variety of shapes and sizes, and they regulate gene expression through a diverse set of mechanisms. Some ncRNAs act as scaffolds, recruiting proteins that modify chromatin structure and silence gene expression. The long non-coding RNA ZIST, which we talked about in the context of X chromosome inactivation, is a good example of that.
So they're like molecular architects shaping the landscape of the genome.
Exactly. Other ncRNAs, like those microRNAs we discussed earlier, can directly target mRNAs, either degrading them or inhibiting their translation.
All right, those tiny assassins, they can shut down gene expression very precisely.
Exactly. And then you have a whole category of ncRNAs that act as guides, directing protein complexes to specific locations in the genome.
Guides. Okay, how does that work?
One example is the CRISPR-Cas system, which you've probably heard of. It's a revolutionary gene editing technology.
CRISPR, right. It's a powerful tool for modifying DNA sequences.
Exactly. And at the heart of that system is a guide RNA. It's a short ncRNA molecule that directs the Cas enzyme, which acts like molecular scissors, to a very specific target DNA sequence.
So the guide RNA is like a GPS system, leading the Cas enzyme to its destination.
Precisely. And once the Cas enzyme reaches its target, it can cut the DNA, allowing for very precise modifications to the genetic code.
It's incredible to think that these ncRNAs are involved not only in regulating gene expression, but also in these cutting-edge gene editing technologies. It really is.
Yeah.
And this is just a glimpse into the fascinating world of ncRNAs. Scientists are constantly discovering new types of ncRNAs and uncovering their diverse roles in regulating gene expression, cellular processes, and even human health.
It's amazing to think that what was once dismissed as junk DNA actually contains a whole treasure trove of regulatory molecules that are essential for life.
It really highlights the complexity and the elegance of the genome.
Okay. We've explored gene regulation from the molecular level to the chromosomal level, discovered those mechanisms that control when, where, and how genes are expressed. We even delved into this world of RNA-based regulation, and now we're seeing the important roles played by non-coding RNAs. It's been an incredible journey, but I have a feeling there's one final layer to uncover.
You're right. We've focused primarily on regulating individual genes and chromosomes. But there's also another level of control, and that's the impact of the environment on gene expression.
The environment. Okay, so we're talking about how external factors can influence how our genes are expressed. Exactly. We've touched on this concept throughout our discussion, but it's worth emphasizing that genes don't operate in isolation. They're constantly interacting with their surroundings, and that interplay can have a profound impact on how those genes are expressed. Think about identical twins, for example.
Identical twins,
Okay.
okay? They have the same DNA.
Right, but their life experiences can shape their health and development in very different ways. And that's partly due to what we call epigenetic modifications. These are changes that can alter gene expression without actually changing the underlying DNA sequence.
So it's like adding annotations to the genetic code without altering the code itself.
Exactly. And one of the key mechanisms involved in epigenetics is DNA methylation, which we discussed earlier.
Right. Adding those methyl groups to DNA can effectively switch genes off.
Precisely. And what's fascinating is that environmental factors, things like diet, stress, exposure to toxins, can actually influence those DNA methylation patterns.
So our lifestyle choices, the things we're exposed to, those can actually change how our genes are expressed.
Absolutely. For example, studies have shown that a mother's diet during pregnancy can impact the DNA methylation patterns in her offspring, potentially affecting their long-term health.
Wow, that's incredible. So our experiences can leave a lasting mark on our gene expression, even across generations.
Exactly. It really emphasizes the interconnectedness of our genes and our environment.
We've covered a lot of ground in this deep dive, from the intricate details of molecular mechanisms to the broader context of environmental influences. It's been a fascinating journey, but I feel like we've only just scratched the surface of this incredibly complex field.
You're absolutely right. Gene regulation is a vast and dynamic field, and there's still so much we don't know. But that's what makes it so exciting. The more we learn, the more we realize how much more there is to discover.
And those discoveries have the potential to revolutionize our understanding of life itself and to lead to new breakthroughs in medicine and biotechnology. Well, for now, I think we've reached a good point to pause our deep dive. But this is definitely a conversation I'm eager to continue. Until next time, keep those brains buzzing, everyone. Okay, so we've journeyed into this amazing world of gene regulation, right? We've gone from these tiny molecules to like entire chromosomes.
Yeah.
We covered all these mechanisms that control when, where, and how genes express themselves, even dived into RNA-based regulation. It's incredible, but I'm still wondering about how all these pieces come together to create this incredibly precise control over gene expression.
That's a great question. It's like asking how individual instruments in an orchestra create this harmonious symphony. The answer lies in this concept of regulatory networks.
Okay, regulatory networks. I'm picturing a complex web, a lot of connections.
Exactly. Think of this interconnected web of genes, proteins, and regulatory elements all kind of influencing each other's activity. And these networks aren't static. They're constantly changing. Right. Adapting to different conditions. They receive input from all these internal and external signals and adjust their output accordingly.
So it's more like a multi-dimensional web.
Yeah.
Right? Not just a straightforward one gene controls another type of situation.
Exactly. And within these networks, you have these things called feedback loops. The output of a process can actually affect its own input.
Like a self-regulating system.
Exactly. And in gene regulation, these feedback loops allow cells to fine-tune gene expression, making sure that the right amount of protein is made at the right time.
Okay, so these feedback loops can either amplify a process or dampen it.
Exactly. You have positive feedback loops and negative feedback loops.
Can you give me an example of how these networks actually work in a real biological process?
Let's consider the cell cycle. You know, that tightly regulated process of how cells divide.
The cell cycle, that's fundamental to life itself.
Absolutely. And when a cell is getting ready to divide, it has to make sure its DNA is replicated accurately.
And those duplicated chromosomes have to be separated correctly into the two daughter cells.
Exactly. And to make sure everything goes smoothly, the cell cycle is controlled by this incredibly complex regulatory network.
Okay, so what are the key players in this network?
Well, at the core of it all, you have these proteins called cyclins, and their partners, the cyclin-dependent kinases or CDKs.
Cyclins and CDKs. Those names ring a bell. Remind me what they're
Think of
about.
cyclins as the on switches for the cell cycle. Their levels go up and down throughout the cycle, and when they're present, they activate CDKs.
And what do CDKs do?
CDKs are enzymes. They attach phosphate groups to other proteins, and that triggers specific events in the cell cycle.
Okay, like a chain reaction.
Exactly. And the activity of these cyclin CDK complexes is very tightly controlled. Feedback loops are a key part of that control.
So how do those feedback loops keep the cell cycle running smoothly?
Well, imagine a checkpoint in the cell cycle.
Okay,
This is a point where the cell takes a pause and checks to make sure everything is going as planned.
like a quality control step.
Precisely. And if there's a problem like DNA damage, a feedback loop gets activated. That halts the cell cycle until the damage is fixed.
Makes sense. You don't want to replicate damaged DNA and pass those errors on.
Exactly. And this feedback loop is often controlled by proteins that inhibit the activity of those cyclin CDK complexes.
So they put the brakes on the cell cycle.
Exactly. One well-known example is the protein P53, a tumor suppressor.
53, right?
When DNA damage is detected, the levels of P53 go up, And that leads to the activation of genes that stop the cell cycle, giving the cell time to repair that DNA damage.
It's like a safety mechanism, preventing the cell from moving forward if something's wrong.
Exactly. And if that damage is too severe to be repaired, P53 can even trigger apoptosis.
Programmed cell death.
Right. Better for the cell to self-destruct than to risk passing on those dangerous mutations. And that's just one example of how feedback loops within these regulatory networks can maintain this incredibly precise control over these complex cellular processes.
I'm really starting to grasp how these networks orchestrate such a beautifully complex system.
It's remarkable, isn't it? This symphony of molecular interactions, each playing its part to maintain harmony within the cell.
And that complexity allows cells to adapt, to maintain their identity, and to carry out all those specialized functions that make life possible. Well, we've explored a vast landscape of information about gene regulation, from the molecular level to the level of these vast regulatory networks, and even touched on the profound influence of the environment. It's been quite a journey.
It has. And while we've covered a lot of ground, the study of gene regulation is constantly evolving. There's always something new to discover. And those discoveries will continue to deepen our understanding of life and potentially lead to new breakthroughs in medicine and biotechnology.
Well, that's the beauty of science, isn't it? It's this never-ending quest for knowledge. And as we continue to delve deeper into these mysteries, who knows what incredible wonders we'll uncover. To our listeners, thank you for joining us on this deep dive into the fascinating world of eukaryotic gene expression. We hope you've enjoyed exploring the intricacies of this fundamental process that governs life itself. Until next time, keep those brains buzzing, everyone.